Good evening. I feel honored to deliver this talk on the discovery of hepatitis E and its impact on global health at AIG Liver Conclave being held at Trident Hyderabad on 30th June 2023. I thank Dr. Nageshwar Reddy and Dr. P.N. Rao and the team for the kind invitation. Hepatitis E is an emerging global disease and a major health problem in endemic regions of the world. My journey in the discovery of hepatitis E has been an incredible human interest story, and this evening I shall uncover it before this august audience. Today is an auspicious day of Eid, and I pray to Almighty to open my chest, ease my task, and untie knots from my tongue so that you understand what I say. I declare a reference to any individual would be circumstantial, and I hold no malice against anybody. The story of discovery of hepatitis E originated in late 70s with my extreme belief in a hidden saga, my passion to explore the unknown, and a golden opportunity of a massive epidemic. I faced challenges of hard weather, difficult terrain, primitive healthcare, an unwilling team to join, health risks to me and to my family, and a lack of funding. I faced these challenges by living in the area and offering care at the doorstep, building a large local team of 500 workers, and spending my personal earnings without any external funding. An innovative door to door survey of affected villages and meticulous recording of the events and thorough analysis of the results were done. Continued surveillance to study the next 10 epidemics and resurveys of the area at 18 months, 14 years, and 30 years formed the hallmark of the study. The decision of visiting and living in the epidemic area was not an ordinary one, as I and all my family members came down with the disease, and today I wish to say to them, thank you for being there with me to support my belief and passion and suffer the pain and anguish in the path of human service and science. To my colleagues and the voice who declined to join me, I say thank you for not joining me so that I could do it alone. Based on the data collected, Kashmir epidemic of 1978-79 was massive and unimodal. The disease pattern was unique. The mode of spread was fecally contaminated drinking water, and the liver histology was distinctive cholestatic. Acute phase sera and fecal samples were non-reactive for acute markers of hepatitis A and B. This led me, a young practicing physician, to believe and announce the existence of a new disease, the epidemic non-A non-B hepatitis caused by hitherto unknown hepatitis virus. The epidemic adversely affected pregnant women, causing a tale of death and devastation. Infections occurred 8 times more in pregnant women than in the control group, which was true for all 3 trimesters of pregnancy. Acute liver failure occurred 16 times more often in infected pregnant women, selectively in the 3rd trimester. Nearly half of the infected 3rd trimester pregnant women became comatose and died, and it was difficult to locate one in the community at the end of the epidemic. This was a remarkable behavior of a unique pathogen and has remained an enigma as of today. The breaking news of the new disease was accomplished in several ways, including the ISG award-winning plenary session paper at Pune, several publications in journals of high impact, presentations at important national and international meetings, and visits and lectures and seminars in centers of excellence. This created a huge buzz and resulted in inquiries, invitations, support, collaborations, from many national and international centers, and visits of many legions to the valley and the epidemic area and to taste the Kashmiri cuisine. Along with the buzz, there was a strong backlash generated to discredit the claims. Skeptics believed that this epidemic was caused by hepatitis A and not non-A, and based their opinion on the extensive studies done on Delhi epidemic 1955-56, which I always believed was a great misstep in the history of viral hepatitis. I faced watch out alert calls and letters from friends and foes, derogatory personal and published letters, one published by Lancet using words like appalled, imagination, and crude and pointless related to the discovery. I fought the skepticism with tolerance, courage, and support, and submitted the sera to several international labs of repute for testing. The results of repeat testing confirmed the non-A, non-B status of the epidemic. So, by 1981, the whole world community was convinced that we were dealing with a new disease and an unrecognized agent. In 1981, I collaborated with Bob Purcell at NIH to perform animal transmission studies on acute phase human samples from Gulmarg Kashmir Epidemic 78-79. Unfortunately, we were not able to inoculate the specimens due to shortage of chimpanzees in his lab till January 1982, a lost opportunity. Soon, in 1983, the world watched with awe an extreme example of self-experimentation by Dr. Balian from Moscow based on his real zest to explore the unknown. This led to the identification of virus-like particles in epidemic non-A, non-B hepatitis. While doing so, Dr. Balian, after consuming 9 fecal samples from Soviet soldiers in Afghanistan, came down with a severe attack of viral hepatitis needing intensive care management. Yet another example of pain and anguish in the path of human service and science. From 1983 to 1987, 2 issues erupted which needed somebody to bell the cat. First, we faced an issue of naming ceremony and a barrage of several proposals and suggestions. I did bell this cat with the alphabet E for all the reasons in the Namakarna. Second, we faced claims of viruses that had not to be from researchers in India, Japan, and France. To counter these claims, 30 investigators met at NIH in April 1987 for a 2 day discourse with advice from 3 legendary rapporteurs. The report heavily depended upon studies from Kashmir, Moscow, and CDC and rejected all other spurious claims. The year 1983 to 1990 were a period of setbacks and frustrations for all concerned as acute phase human samples contained an extremely low amount of virus-like particles, not sufficient for cloning and sequencing. A surprise observation of finding large quantities of virus-like particles from bile and not from stools of infected macaques ended the dilemma. Soon, based on construction of cDNA libraries, finding of a clone ET1.1 of 1.3 kilobase from infected macaque's liver by the gene lab hit the bull's eye and subsequently led to the cloning and sequencing of the full-length 7.2 kilobase Burmese strain of the hepatitis E virus. Over the 44 years journey, our team at Kashmir traversed 3 eras of ambiguity, hope, and hype in hepatitis E research. Based on the activities of the field survey team, the liver unit, the liver lab, and the collaborations, we investigated 10 epidemics of hepatitis E, studied thousands of sporadic hepatitis and acute liver failure, and faced an ever expanding pool of neonatal hepatitis E. Several landmark studies were conducted and published in journals of repute to understand the biology of the pathogen, the hepatitis E virus, and the manifestation of the human disease, the hepatitis E. Apart from discovery, we established 5 clinical forms of hepatitis E, 4 routes of transmission, long-term protective antibodies following natural disease, and finally, the enigmatic, expanding, and complex relationship of hepatitis E and pregnancy to understand the pathogenesis of higher mortality. Over the same 44 years, based on the global data, many milestones have been crossed to understand the structure, the biology, and diversity of the agent, habitat and host of the agent in the animal kingdom, evolution and population dynamics over time, existence and changing behavior of the pathogen beyond our borders, and discovery of a highly effective vaccine, 239, a great step to ultimate cure and control of this pathogen. Hepatitis E virus has unique structure. Hepatitis E virus is a positive sense single-stranded RNA virus of 7.2 kilobase, icosahedral in shape with 2-3-5 symmetry, with a cap, 2 UTRs, one each at 5' and 3' end, 4 cis-reactive elements forming the stem loops across the genome, and 3 partially overlapping open reading frames with defined functions. The native virus has a triangulation number of 3. 3 unique features of the virus include a quasi-enveloped hepatitis E virus circulating in the blood, a phenomenal 3-domain crystal structure of capsid with P2 domain as a dimer, and exceptionally important protein expressed from ORF4 in genotype 1 alone. The protein of ORF4 is 124 amino acids, promotes replication of the virus, is indispensable for the life cycle of the genotype 1, and is possibly involved in the pathogens of increased severity in pregnancy. Hepatitis E virus has earned the status to be the sole member of a new family of viruses, the Hepaviridae, with remarkable heterogeneity. Hepatitis E virus is divided into 2 subfamilies, 5 genera, and 10 species, and infects over a dozen of hosts in the animal kingdom. Other than causing several clinical syndromes in chicken, important avian population, the virus does not cause any clinical illness in any other animal host it inhabits. Humans are infected by 4 genotypes in the genus Orthohepativirus A, genotype 1 and 2, or anthropognotic, and transmitted from humans to humans alone. Genotype 3 and 4 are enzootic and infect pigs and few other animals, and infect humans through peculiar foodborne zoonotic transmission. Few human infections in solid organ transplant patients have been reported to occur from camels in Dubai by eating and drinking milk, camel milk and meat, and from rats in Hong Kong through exposure to droppings in the environment. The path followed by the virus from binding to egress from hepatocytes is depicted. Hepatitis E virus life cycle remains poorly understood as of today, and there are large knowledge gaps in receptors and functional domains of ORF1. The distinct entry pathway of non-enveloped and quasi-enveloped virus particles are shown. While quasi-enveloped particles circulate in the blood and are immune evasive, the particles lose coat in bile ducts and circulate as naked particles in the environment. To commemorate the political and scientific controversy around the evolution of SARS-CoV-2, the evolution and population dynamics of human hepatitis E virus are shown. For a sigh of relief, humans seem to be farther from bats and chicken and nearer to rodents to evaluate cross-species transmission. Around 5 to 13 centuries before the present, human hepatitis E virus has evolved through natural selection into anthropogenic genotype 1 and 2 affecting humans alone, and enzootic 3 and 4 affecting pigs and related animals and transmitted to humans through peculiar foodborne pathway. The spread of hepatitis E genotype 4 to Japan coincided with the export of pigs from India. Genotype 4 underwent a recent expansion to China to nearly replace the long existing genotype 1. Hepatitis E has a global distribution. Genotype 1 and 2 infect humans alone with contaminated water as the transport vehicle and cause hyperendemic and endemic disease in poor resource countries because of poor sanitary disposal based on practice of open defecation. In contrast, genotype 3 and 4 infect and exist heavily in swine population, and autochthonous human infections occur by peculiar habit of eating raw or undercooked pig meat, liver, and sausages. So, clean water and eating cooked pig meat are major weapons to fight against and eliminate hepatitis E. This slide depicts the pictorial mode of transmission of hepatitis E. In endemic areas, there are unique ways in which public water supplies are contaminated by sewage. However, open defecation continues to be a major public health issue in the Indian subcontinent. In industrialized countries, cross-contamination of the virus across deer, boar, and pig occurs, and exposure to human follows several pathways as shown. Hepatitis C virus is an emerging global infection of major public health importance. One third of the world population is infected, with an estimated 14 million cases, 300,000 deaths, and 5,200 stillbirths annually. Genotype 1 and to a lesser extent genotype 2 are major public health problems in endemic areas of the world through repeated massive waterborne epidemics, each involving hundreds and thousands of cases. Hepatitis E in these regions is the most common cause of acute sporadic hepatitis, acute liver failure, and acute and chronic liver failure. There are enormous load of hepatitis C in pregnancy and fetal and neonatal hepatitis C infections. Of late, there are declining trends in these figures due to improvement in the sanitation in India. Hepatitis E is no more a disease of those third world countries. Genotype 3 and 4 infections are the most successful zoonotic viral disease in human history and cause zoonotic foodborne autochthonous infections in industrialized countries. With the availability and application of hepatitis C virus diagnostics, the existence of hepatitis C in several other forms has been established, including transfusion-transmitted infections enforcing screening programs by several European countries, a large pool of genotype 3 chronic liver disease in solid organ transplant, HIV and hematopoietic neoplasms, drug-induced liver disease, and a series of syndromes caused by extrahepatic manifestations of hepatitis E virus. As shown by us and others, hepatitis C in developing countries leaves behind a tale of death and devastation in pregnant women. This is true in epidemics, sporadic viral hepatitis, and acute liver failure. Acute liver failure in pregnancy is an explosive disease with unique clinical features. Obstetric complications and neonatal hepatitis C virus infections are frequent, affecting the maternal and fetal health status. The pathogenesis of higher morbidity and mortality due to hepatitis C virus infection in pregnancy is complex and remains to be fully understood. Earlier studies have revolved around hormonal imbalance, immune dysregulation, and a poor nutrition status of pregnancy. There have been some exciting developments in the recent past. First, this adverse relationship is genotype specific and restricted to genotype 1. Second, genotype 1 alone has an additional ORF4 which expresses a protein that promotes viral replication and consequent higher liver injury. Based on protein for ORF4 power, genotype 1 infects placenta, causing necrosis and apoptosis, leading to obstetric events and release of cytokines in the mother's blood, enhancing liver injury. In addition, it breaks the maternal fetal interface, leading to vertical transmission, fetal infections, severe fetal disease, and fetal deaths. Fetal deaths promote liver injury in the mother and DIC. The development of accurate diagnostic assay for the detection of hepatitis E virus infection remains challenging, and many outstanding issues remain. The serological tests have issues of sensitivity and specificity of assays. NAT testing, essential in several scenarios, is available in specialized labs only, and these tests need to be developed, standardized, and made available widely for a day-to-day practice. Apart from modulating immunosuppression, ribavirin monotherapy has been the cornerstone of managing chronic hepatitis E in solid organ liver transplant patients. Pegylated interferon alpha and sofosbuvir or alternative options in case of ribavirin treatment failure. The use of ribavirin is contraindicated in pregnancy. However, its use in hepatitis E virus acute on chronic liver failure is worth a trial in selected cases. The discovery of hepatitis E vaccine, the HEV-239 Heculin, has been a breakthrough in the history of hepatitis E and has been accomplished through expression, virus assembly, and formation of highly immunogenic virus-like particles from 239 amino acids of ORF2 protein expressed in the E. coli insect cell system. The vaccine has successfully completed phase 2 and phase 3 trials and is highly efficacious with the long-lasting protective effects. Its availability and application in other regions of the world, particularly in Indian subcontinent, is being actively pursued. What are our challenges in the control and cure of this global human pathogen? These include further developments, standardization and availability of diagnostic tests, better understanding of perplexing biological and epidemiological behavior of the pathogen, development of treatment strategies and management policies, and develop and implement effective preventive strategies, especially the hepatitis E vaccines. Ladies and gentlemen, I conclude and submit that the path traversed by me in the discovery of hepatitis E has been a fabulous journey, and I was blessed to be on board for a wonderful trip. The journey envisaged hard work, persistence, belief in oneself, and honesty of purpose. It meant spending days on snowbound, dilapidated roads of those villages and feeling the pain and anguish of the sufferers. It needed courage to stand on one's feet and face the inherent bias, skepticism, and listen to the voice. I spent nights staying awake with pen and paper to write that what I believed was true and to uncover. The journey was treacherous, hard, and full of failures and some successes. However, the end has been pleasant and rewarding, for discoveries do not come without a price to be paid. Thank you for your patient hearing.